The most prevalent molecule is water. It is present in every cell, no matter the type. Linus Pauling was a scientist who taught at Caltech. He taught chemistry, and he looked at the periodic table of elements. He found that the right-hand side of the periodic table tended to be more electronegative than those on the left-hand side. The right-hand side features more of the solids, while the left-hand side features the gases. Oxygen is more electronegative than hydrogen. It has a greater pull on electrons than hydrogen. Electrons spend more time around oxygen than they do hydrogen. Therefore, the water molecule is polar. There are two types of covalent bonds. We have polar and nonpolar bonds. Polar bonds are the unequal sharing of electrons, while nonpolar bonds share the electrons equally. A hydrogen bond is an electrical attraction of a hydrogen that is bound to an electronegative atom with another electronegative atom. Opposite charges attract. Hydrogen bonds are the most common bonds found in living organisms. Linus Pauling's mistake was that he thought hydrogen bonds hold DNA together, but they don't. Hydrogen bonds are weak and can easily be broken. They are about 23 kilojoules per mole to break in water. Covalent bonds are strong. It takes about 470 kilojoules per mole inside an OH bond. If two atoms have the same or very close electronegativity, they will form nonpolar covalent bonds. The greater the difference in electronegativity, the more polar the bond. For example, a carbon-carbon bond is about 348 kilojoules per mole required to break. Hydrogen bonds hold proteins in their shape. This is absolutely necessary for their function. Dipolar molecules can act as a polar solvent. Aqueous chemical factors, they can dissolve, such as polar molecules and ionic molecules, because they are hydrophilic. Nonpolar molecules include lipids. These do not dissolve in water. They are hydrophobic. For example, oil and vinegar, they separate in a mixture because vinegar is a weak acetic acid. The nonpolar hydrophobic substance, in this case oil, will not dissolve in a polar solvent. Polar and ionic molecules are both charged. For example, sodium chloride. Sodium is on the left-hand side of the periodic table, and it has one electron in its outer orbit. Chlorine is on the right-hand side of the periodic table, and it has 7 electrons in its outer orbit. The electronegative difference is so great that chlorine will pull 1 electron completely away from the sodium and add to its own orbital to equal 8 electrons. Now, sodium has 1 more proton than electron, so it has a positive charge. Chlorine has 1 more electron than proton, so it has a negative charge. The negative chlorine ion makes an anion called the chloride ion. Sodium, with its positive charge, is now a cation. Sodium chloride grouped together forms ionic bonds, and together they cluster to form a crystal lattice, formerly known as salt. If we pour water over this salt block, it will pull ions off of the salt block. More water than salt means that the water surrounds the individual ions, forcing the positive and negative charges apart and surrounding them. They are pulled into solution. Water in cells works the same way on all water-soluble molecules in cells. Some of these water-soluble molecules in cells include carboxyl groups, amines, phosphates, hydroxyl groups. Nonpolar molecules. Amphipathic are very important. It means they are both polar, hydrophilic, and nonpolar, hydrophobic. There is a polar head and a nonpolar tail on each of these amphipathic molecules. When they meet water, the head is attracted and the tail orients itself away from the water. This forms micelles. Here, in this 3D structure, Nonpolar tails minimize the interaction with water by clustering together. The hydrophobic interaction is similar to hydrogen bonds and ionic bonds. It is weak and it is designed to achieve thermodynamic stability. Examples of these include proteins, sterols, phospholipids, and fatty acids. The hydrophobic interaction forms the basis of membranes. The van der Waals interactions play a critical role in structure of biological molecules. It features two atoms in very close proximity to form an electron cloud interaction. These are minor interactions. An interaction in a biological molecule is primarily due to the existence in an aqueous environment and is greatly influenced by water. A solution is a two-component mixture. It is composed of a solute, which is the stuff that is dissolved, and a solvent, which is what does the dissolving. Pure H2O, pure water, contains colligative properties, means that properties of water can change when you're comparing pure water to a solution. Solutions lower the concentration of water. Semi-permeable membranes allow water, but not the solute to move through. For example, if we have pure water on the left-hand side, separated by a semi-permeable membrane, and a solution on the right side, when we have a greater concentration of water on the left to a lesser concentration of solution on the right, then we see a movement of water across the membrane known as osmosis, and it produces osmotic pressure as the greater concentration of water moves to the lesser concentration in the solution. This describes the cell environment. Water molecules move from the greater concentration to the lesser concentration. If the movement from the solute inside the cell to the solute outside of the cell is greater, we have a hypotonic solution. If the concentration of solute inside the cell is less than the concentration outside the cell, it is hypertonic. If both concentrations are equal, it is isotonic. If we assume the concentration of solution inside the cell equals the concentration of solution outside the cell, we have an isotonic. If we assume the concentration of solute inside the cell is equal to the concentration of the solute outside the cell, it is called isotonic, where the cell neither gains nor loses water, and there is no net change in the water flow. The cell is hypertonic if the cell loses water and shrinks. This is called plasmolysis in plants or cremation in animals. This is when the concentration of the solute inside the cell is less than the concentration of the solute outside the cell. Here, the water flows from inside the cell to outside the cell. The cell is termed hypotonic when the net flow is from the outside to the inside of the cell. The cell swells until it explodes, or lyses. The cell swells until it pushes against the cell wall. The water, which is in the central vacuole, expands in plants. Pressure pushing on the cell wall equals turgor pressure. Cells like hypotonic conditions. They go to great lengths to maintain water in the cell. This is when the concentration of the solute inside the cell is greater than the concentration of the solute outside the cell. If the cell becomes dehydrated, physiological saline IV drips, such as an isotonic solution, can help avoid swelling. It limits the amount going in until you begin to hydrate. Every once in a while, water has a tendency to ionize or dissociate. This is when water breaks into its hydrogen cation and OH anion, or it forms a hydronium ion, which is H3 plus O, and an OH negative ion. The formation of a hydronium ion is more common, and this process is reversible. This is critical to cellular function and it is important that we as scientists need to quantitate it. Chemical equilibrium is when both the reactants and the products are at equilibrium, and there is no net change in concentrations. We can represent chemical equilibrium using the equilibrium constant, K equilibrium. Note that this does not mean that the concentration of reactants A plus B equals the concentration of products C plus D. This is at the point at which A plus B goes to C plus D, and C plus D goes to A plus B. Our equilibrium constant KEQ is equal to the concentration of the products over the concentration of the reactants. In this case, the concentration of C multiplied by the concentration of D, all divided by the product of the concentration of A multiplied by the concentration of B. If we're looking again at a water molecule, 1 in every 500 billion molecules dissociates. For pure water at 25 degrees Celsius, also known as room temperature, the KEQ can be represented as the concentration of H plus ions multiplied by the concentration of OH negative ions divided by the concentration of water. We will represent this in grams per liter divided by grams per mole, and when divided it gives us moles per liter, also known as molar. For water, this value is 55.5 molar. Another equilibrium constant is known as Kw. This is the ion product of water, and it forms the basis for the pH scale. The Kw is the KEQ for water, and the Kw is equal to 55.5 molar. The KEQ equals 1.8 times 10 to the negative 16 molar at 25 degrees Celsius. Kw is a constant. If the concentration of hydrogen ions increases, then the concentration of hydroxide anions decreases. If the concentration of hydroxide anions increases, then the concentration of protons decreases. We can always calculate the concentration of H plus if the concentration of OH minus is known. Similarly, we can always calculate the concentration of OH minus if the concentration of H plus is known. That is why Kw forms the basis of the pH scale, as the concentration of H plus in an aqueous solution will be between 1.0 molar of H plus and 1.0 molar of OH minus. The pH, by definition, is 1 divided by the log with base 10 of the concentration of hydrogen ions. This is equal to the negative log base 10 of concentration of hydrogen ions. A neutral solution, such as pure water at 25 degrees Celsius, is where the concentration of hydrogen ions equals 1 times 10 to the negative 7 molar. Then the pH equals negative log of 1 times 10 to the negative 7, giving us log of 1 plus log of 10 to the power of 7, which equals 0 plus 7. Therefore, a neutral solution, pure water at 25 degrees Celsius, has a pH of 7.0. This value is not arbitrary, and it is derived from the Kw. A pH greater than 7 is called alkaline, or basic. This is when the concentration of OH minus is greater than the concentration of H plus. A pH with a value less than 7.0 is known as acidic. This is when the concentration of H plus is greater than the concentration of OH minus. Please note that the pH scale is logarithmic. A pH 6 versus pH 5, for example, shows a difference of 1 times 10 to the minus 6 molar versus 1 times 10 to the negative 5 molar. The pH scale is the most commonly used measurement in biochemistry. The physiological pH is somewhere between 6.8 and 7.2. For example, the stomach pH is acidic at a value of pH 2. Enzymes are best suited for the pH environment. pH affects enzyme activity. pH affects macromolecular structures. They prefer neutral pH because their shape is critical for function. pH is also used for medical diagnoses. The blood pH is important for regular function. DNA work is done at a specific pH. Acids, bases, and buffers. Strong acids or bases completely ionize an aqueous solution. Biologically, we want to avoid strong acids and strong bases. In these cases, such as a hydrogen chloride molecule, the arrow goes only in one direction when they ionize. It will only go towards H+ and Cl-. This is a strong acid. Sodium hydroxide, NaOH, is a strong base, and this dissociates into Na+ and OH-. An acid is a proton donor, biochemically, and bases are proton acceptors, biochemically. Weak acids form an acid-conjugate base pair and do not completely dissociate. These are preferred biochemically. An example of a weak acid is acetic acid. This can dissociate into H+ and an acetate ion, which is the conjugate base to acetic acid. This process is reversible. In every case of a weak acid, the weak acid will dissociate into an H+ ion and its conjugate base, and the reaction will always be reversible. The tendency for weak acids to lose its proton is governed by the KEQ, the equilibrium constant, represented as the H+ multiplied by the conjugate base divided by the weak acid. This is also known as the Ka. The Ka is the ionization constant, or the dissociation constant. Strong acids have larger Kas in general. Therefore, the Ka is a good indication of the strength of a weak acid. We can also represent the Ka by its pKa, which is 1 divided by the log base 10 of the Ka, or negative log base 10 Ka. The stronger the tendency for the proton to dissociate or produce H+, the stronger the acid and the lower the pKa. We can use titration to find the pKa. This is the measured volume of acid titrated with a known concentration of strong base. It should be added in increments and can be used to measure the pH. Small changes in pH create large changes in biological processes. As we already mentioned, the physiological pH is between 6.8 and 7.2, but neutral is preferred biologically. Changes can result in cell death, so the body must maintain a constant pH. The body uses natural buffers. Buffers are an aqueous system that is resistant to pH changes upon addition of small amounts of acid or base. The weak acid or conjugate base pairs are most commonly used as buffers. Each molecule has a characteristic zone where it acts as a buffer. Each buffer will have a buffering capacity, which is the range at which it buffers. We can represent the pH is equal to the pKa plus the log of the concentration of proton acceptor divided by the concentration of proton donor. This is the Henderson-Hasselbalch equation, and it fits the titration of all weak acids. When you're considering H2O as aqueous solutions, the cell is, in essence, 70 to 90% water. Therefore, it is an aqueous solution. If we removed all water from the cell, we would leave about 10 to 30% of the cell, and this is just dry weight. The cell has atoms in dry weight. Its 4 major elements are carbon, hydrogen, nitrogen, and oxygen, where carbon accounts for about 50%. There are secondary elements, phosphorus, and sulfate. There are a mess of trace elements: calcium, potassium, magnesium, sodium, iron, silver, gold, iodine. But about 50% of the dry weight is carbon. Carbon has 6 protons and 6 electrons. Of those 6 electrons, 4 electrons are in the outer orbital. Carbon has a tetrahedral structure, and it is able to form 4 covalent bonds. It forms stable covalent bonds with 3 other major elements. Single and double covalent bonds are possible with other carbon atoms, as well as other atoms such as nitrogen. Carbon with a triple bond to nitrogen is termed cyanide, and this is not great for living systems. There are infinite possibilities for carbon compounds. The number of bonds and type of bonds between carbon molecules plays an important role in resultant molecules. It can change the properties. For example, a single bond between carbon and carbon allows for free rotation. A double bond between carbon and carbon is shorter than the single bond, but it is more rigid and no rotation is allowed. Therefore, it creates a more defined structure. Carbon molecules can form linear chains or branched chains or cyclic rings. Carbon can form covalent bonds with hydrogen, oxygen, nitrogen, and other elements. It can form functional groups. For example, coal is nothing more than carbon, and diamonds are compressed coal. If we put it in water, nothing happens. Straight carbon molecules are not useful for biological activity. Functional groups are useful for biological activity, as they are soluble in water. Some common functional groups include a hydroxyl group, amino groups, carbonyl carbon groups, carboxylic acids, sulfhydryl groups, phosphate groups, and others. Hydroxyl groups, when added with carbon, form alcohols. Amino groups, or amines, are organic bases. There is a carbonyl carbon, which is a carbon double bonded with oxygen. If it is at a terminal end, it is known as an aldehyde. If it is internal, it is a ketone. Carboxylic acids are organic acids, and they contain our carboxyl group, which is carbon double bonded to oxygen and single bonded to a negative oxygen ion. SH is a sulfhydryl group, also known as thiols. Adding this group to a molecule gives it a distinct odor. Phosphate groups are important for energy transfer. Most functional groups are polar. Biological molecules exist in aqueous solution and can have more than one functional group on the same molecule. Amino acids contain an amine and carboxyl group. Molecules and cells. We can use whatever cell. Source does not matter. Within that cell, we will find the same about 100 to 200 small organic molecules in every cell. They serve many different functions, and they represent central metabolites. They occur in major chemical pathways. They represent building blocks of larger molecules found in the cell. They are used in the construction of cellular macromolecules. The building blocks are amino acids, simple sugars, nucleotides, and carboxylic acids, or fatty acids. They have been conserved throughout evolution, and they are found in the cell. Most macromolecules represent polymers. High molecular weight molecules vary in the number of building blocks used in construction. Synthesis requires astounding amounts of energy. Most of the cell's energy is used. There are 4 major types of macromolecules. The first are polysaccharides, also known as carbohydrates. These are polymers of simple sugars, or monosaccharides. Their functions within the cell include energy storage and structural mechanisms. There are oligosaccharides, which serve as lipids or proteins that compose glycolipids and glycoproteins. These are found on the surface. They play a role in cell signaling and cell-to-cell recognition. For example, tissue typing is used in the medical industry. This is where organs must match as much as possible for the organ transplants. Number 2, lipids. These are fats, oils, and sterols. These are the only macromolecule that is not a polymer. They are hydrocarbon derivatives. They serve as membrane components. These are energy-rich storage molecules. They can form pigments, and they act as intracellular signals. Number 3, proteins. These are the second most prevalent molecule in the cell. They compose the largest fraction of a molecule in the cell. These are polymers of amino acids. They vary greatly in length. Proteins are the most diverse class of macromolecule. Their functions include enzymatic, structural, signal receptor, and transporters. When functioning as an enzyme, they are responsible for catalytic activity in the cell. This is when chemical reactions are sped up. Not all enzymes are proteins. Some can be nucleic acid enzymes, such as RNA molecules, which have catalytic activity. Proteins as structural elements make up parts of the cell membrane and internal membranes of eukaryotic cells. As transporters, proteins move molecules across the membranes, in and out of the cell. Number 4, nucleic acids. Nucleic acids are polymers of nucleotides. These are small molecules in the cell. RNA and DNA are 2 types of nucleic acids, as they have different nucleotides. They have 3 major functions: transmission of information, storage of information, and retrieval of information. Everything you are or ever will be is because of DNA. Nucleic acids also have a structural and functional role in supramolecular structures. For example, ribosomes, which is the site of protein synthesis, is made up of RNA, and spliceosomes, which are utilized in the removal of extrons and connections of introns in DNA splicing. In each of the 4 different classes, key to the structure and function is their 3D shape. This is due to the molecules that make them up, their structure, and their functional groups. Stereochemistry is the 3D arrangements of atoms and molecules. Configuration is the fixed spatial arrangement of atoms, and it depends on the presence of double carbon bonds, which allow for no rotation, and a chiral center, which is a carbon atom with functional groups attached in a specific order. Carbon molecules are so diverse, with various bond patterns and functional groups. Many exist as stereoisomers, which are molecules with the same chemical bonds but having a different 3D arrangement of atoms spatially. An isomeric form is very important in biochemistry. Configurational isomers cannot be interconverted without at least temperature-breaking covalent bonds. If the functional group is on the same side of a double carbon bond, it is known as cis. If a functional group is on opposite sides of the double carbon bond, this is known as trans. Both cis and trans are geometric isomers. Each isomer is well defined and has its own specific chemical properties that get acted upon by enzymes. The orientation around a carbon-carbon double bond is critical to biological function. The chiral center is the carbon atom to which 4 different functional groups are attached. If the arrangement of the atoms are different around this chiral center, they are known as stereoisomers. Stereoisomers that are mirror images are known as enantiomers, but they are not superimposable. Non-mirror stereoisomers are known as diastereomers. Chiral centers are usually present in organics in one of 2 isomeric forms, the D, which is the right, and the L, which is the left. Therefore, we have L amino acids and D sugars. Life evolved in asymmetric manner, and life is asymmetric. The configuration is a fixed spatial arrangement of atoms and can only be changed by temporarily breaking covalent bonds. Conformation is the spatial arrangement of functional groups. The 3D structure is the configuration and conformation, and the compound of both conformation and configuration. The most abundant macromolecule in the cell are proteins. Proteins are constructed from the same 20 amino acids. This is true for every protein in every organism. The difference is the linear composition or sequence of amino acids. Amino acids are molecules in which we have a carbon atom attached to an amino group and carboxyl group and H and R group. The R group is the side chain and can differ in size, structure, and electrical charge. This affects its water solubility. Some amino acids don't like water and are therefore insoluble. Others adore water and will fully go into solution. An exception to the basic structure for amino acids is proline, which has an R group and an amine group joined together in a ring. Frederick Sanger wanted to sequence proteins, and he chose insulin. He wrote up the name of every amino acid in the chain and found that there were about 30. As more proteins were sequenced, more amino acids used 3-letter abbreviation because it was more expensive. 1-letter abbreviations are now commonly used. Examples of amino acids and their 3-letter and 1-letter nomenclature include alanine, ALA, A, phenylalanine, PHE, F, proline, PRO, P, threonine, THR, T, tyrosine, TYR, Y, valine, VAL, V, glycine, GLY, G. Glycine is different than the other 20 amino acids. There is a convention for how to draw amino acids. Everything is drawn as they appear in the cell. By convention, the amine group is drawn on the left for L and on the right for D. Proteins are constructed from L amino acids. Enzymes catalyze stereospecific L amino acids. Each protein has its own characteristic 3D shape. The linear sequence of amino acids differs, and their R group differs. Characteristics of proteins are determined by R groups, and amino acids are classified by their R groups. The 5 categories are nonpolar, aliphatic, hydrophobic. Number 2, aromatic. The side chain has a ring structure. Number 3, polar uncharged. The R group has something to increase water solubility. 4, a polar positive charge. The amine group in the side chain acts as a protein acceptor. This is basic in nature. Number 5, the polar negative charge. The carboxyl group is in a side chain and is a proton donor. These are acidic.